So Beatrice is actually Professor of Pharmacology and Pharmacotoxicology at Lisbon University. And she's also the chair of the IMI Scientific Committee, so we know Beatrice very, very well. And we're now going to have a session looking at an area where there are many questions, and that's in terms of the regulatory challenges that we face in the sort of drug-food continuum and the microbiome. So I'll hand the floor over to you now.
Thank you. Thank you, Hugh. So good morning. So here we are on our session for regulatory challenges. We have two speakers, Professor Yolanda Sanz and Dr. Nico Mihokovic, from representing both sectors, so in their regulatory perspective on food and medicines component. They will address specific aspects and more general aspects in this in their 5-minute talk: how to regulate microbiota-directed foods, the regulatory issues surrounding the gut microbiota, the challenges and opportunities for collaborative research, and very interestingly, how to deal with, let's say, microbiota from both components in terms of, I would say, personalized health care. So I would start by asking Professor Yolanda Sanz to start her talk. She will give us the the perspective, the regulatory perspective from food aspects. Professor Yolanda Sanz, she is a PhD in pharmacy, professor of research of the Spanish National Research Council. She's head of research unit on microbial ecology, nutrition and health, and has been previous assistant professor on human dietetics and nutrition. Regulatory component, she's a member of the panel of the Dietetic Products, Nutrition and Allergies of the European Food Safety Authority, EFSA, and she has been vice-chairing that panel up to 2015, and her scientific interests are in the field of human microbiome and influence on progressing from health to disease. And she coordinates research project, in particular a large one, which is very international, called Micro New Gut. Thank you, Yolanda, for your presentation.
Well, good morning, everybody, and thanks a lot for the introduction and also for the invitation to participate in this interesting forum. Well, in my turn, I'm going to talk about regulatory challenges in this field, and to do so, I will do it on the basis of my own personal experience as a scientist that is working in this area, as many of you here, and also on the basis of the experience I gained evaluating health claims and novel foods as part of the EFSA panel during the last years. Although I also must say that here I'm not representing EFSA, and what I'm going to do is to try to give you my personal vision. Well, to do so, what I'm going to do is to recall the main principles of the health claims regulation, that is the one that more closely affects this field. I will also explain to you how the EFSA panel tries to reconcile science with regulation in the context of health claims. I will go through the promises of the microbiome in the drug-food continuum, and finally, I would like to tell you also how EFSA is taking action to face the new challenges that are coming from this field. Well, the regulation on health claims entered into force in July 2007, already 10 years ago, with the main purpose of providing higher level of protection to consumers. In such a way, they were protected, for example, from misleading claims, and also with the idea of helping the consumers make healthier food choice in their daily life and promote their long-term health in the end. But this regulatory text also harmonized the procedure to authorize and use these messages that imply a relationship between a food and a health benefit, and that reach the consumers in many different ways, and in this way ensures also fair competition among the different business operators. And there is also a section that tries to promote innovation by offering 5 years protection to some of the applicants that could provide proprietary data that they generate exclusively for this application. But well, I must say also that this health claims regulation depends on the European food law, and according to which foodstuffs cannot be attributed the property of preventing, treating, or curing diseases. This is the way to differentiate between foods and drugs, and this has enormous consequence on the type of claims we can make, and also in the way we formulate claims. This means the wording we use when making claims. And well, we can make function claims that are those related to the role of a food in the maintenance of a body function or in the improvement of a body function when there is physiological decline. And we can make disease risk reduction claims referring to the ability of a food to reduce a risk factor for the disease. And well, in fact, for the purpose of communicating the health properties of foods to the consumers, in principle, subjects with a disease cannot be the target of a claim. The target of the claim should be general population, healthy general population, or subgroups of the general population. And this introduces an important constraint because it's very difficult to prove the efficacy of a food in healthy people. It's like to add health to health. On the other hand, function claims cannot refer to a disease, and disease risk reduction claims cannot refer to the reduction of the risks of the disease, but should refer to the risk factor. And here we have another limitation also. It's the fact that there are few risk factors that have been considered validate. One is high cholesterol levels, high blood pressure, and the presence of real pathogenic microorganisms that can be toxigenic Clostridium difficile. And well, much work should be done in the identification and validation of other biomarkers that facilitate to make and prove this type of claims. But well, in the last guidance on health claims that was published by EFSA last year, it was already clearly recognized and stated that this regulatory framework many times may be in contradiction to the scientific principles we have to apply during the evaluation process. And even if the target population of the claim should be general population and healthy population, the study groups that are used to prove the efficacy of the foods or a food ingredient in some cases have been diseased subjects, when we expect that the same phenomena, the same effect occurs also in subjects without the disease. We have also has appropriated study groups subjects with diet-related disorders such as obesity, subjects with hypercholesterolemia or hypertension, because we recognize clearly that these study groups are the ones who could benefit the most for these claims and these dietary strategies. In fact, diet-related diseases are managed by diets in many cases. And we also indicate that the relationship between a food constituent and a function of the body many times can be best measured using disease outcomes, and also that disease outcomes provide stronger evidence than risk factors on the ability of a food to reduce the risk of a disease. And that's obvious from a scientific point of view. And well, these are just examples of the cases in which we have applied this approach, and we have included these study groups in the overall assessment of the evidence. For example, we have used study groups IBS patients to prove an effect in gastrointestinal discomfort, because gastrointestinal discomfort is also often occur in general population. The difference is the intensity and the severity of the symptoms, but we could do this extrapolation. We have also considered appropriate study groups, for example, those who have mild hypercholesterolemic levels, to prove an effect of a food in the reduction of LDL cholesterol. And we have also used clinical outcomes such as symptoms of infections, incidence of infections in claims related to the ability of a food to increase our defense against pathogens. But well, the problem here is these are like exceptions to the rules. And this depends on an evaluation, a scientific judgment that is done by the panel on a case-by-case basis. And the problem here is that there is a certain degree of uncertainty for the applicants in relation to whether the study population, the design of the study, and the outcome variables are going to be finally accepted during the evaluation process. And they need to be, to some extent, sure when planning the studies and when designing all the protocols that are going to be part of the dossier to be submitted to EFSA. And well, this is also one of the reasons why we can find some contradictions between the claims that have been approved in the food area and the messages that we can find in some professional guidance about the applications of probiotics. This is just an example published in 2011 of the World Gastroenterology Organization indicating that some probiotics could be useful for the treatment of acute diarrhea, for the treatment of inflammatory bowel diseases, and so forth. And in fact, these type of claims are not eligible in the food area, and are in fact the risk managers the one who have to decide whether a claim is a food or a medicinal claim. But well, it's true that the relative lack of success in getting health claims approved in our area, and particularly in the probiotic field, contrasts to the increased interest in the microbiome research area, the number of very, very good papers that are being published in this area, the promises that we are generating all in the field. And in fact, with the evidence we have so far, we can conclude that there are clear associations between alterations in the populations that inhabit different parts of our body, particularly the gut, and many diseases, mainly diet-related diseases and chronic diseases such as obesity, type 2 diabetes, or cardiovascular diseases. And more recently, there are also links being established with neurodegenerative disorders and neuropsychiatric disorders that are major burdens in our society. So if we start thinking that perhaps the microbiota could be at the root of the disease, could be a causal or contributing factor to the disease, we could start envisaging strategies that could help us to prevent disease at early stages of development, and also perhaps to intercept and arrest disease before the disease symptoms appear when we are in this period of suboptimal health. And well, there are already some applications that Dusko has mentioned, and as explained very well. One is feeling really well in the drug area is the use of fecal transplants. This has been considered a strategy of high rate of success in the treatment, for example, of recurrent Clostridium difficile infection. But well, we can also think of other alternatives that are really fitting in the food area as well. And for example, we can think of cocktails of bacteria from our intestinal gut, from healthy subjects that can help us to replenish those bacteria that we have lost because of unhealthy dietary habits and are putting us at risk of disease. We can also think about dietary strategies a bit more sophisticated than the classical prebiotic likes to also contribute, for example, to increase the diversity of our gut microbiota that in principle seems to also protect us against disease. And we can also think about products that are produced from these bacteria, and that could also provide health benefits such as short-chain fatty acids, proteins, and so on. Well, I would like to mention how EFSA is taking action in this area and is trying to be ready for what is coming to the panel for evaluation in the future, in the next years. And in this regard, for example, I would like to recall the EFSA guidance on novel food applications that was published last year as a consequence of the new regulation in novel foods, where we address how you have to evaluate the safety of new microorganisms that have not been used for food in humans so far, and they don't have a QPS status. I will not go into detail because this is not the purpose of this meeting and this is published, but what I want to tell you is that this is a reality already. And we have already approved as novel foods a strain of Clostridium butyricum and milk products fermented with Bacteroides seal and solvents. Well, I would like to draw your attention also on another EFSA guidance that was released before the summer, has been under consultation until 15 September. This was prepared by the Fecal panel and includes all details of the characterization of the microorganisms in terms of safety that you could also follow in the context of those that are going to be in the process of being evaluated as novel foods. This guidance is very much detailed, and in this guidance, not only the Feed panel has participated, but also the GMO panel and also the NDA panel. Well, another new challenge that is coming to us is this trend to develop personalized solutions. And well, in this case, this is not in place yet. We don't know where in the regulatory text this is going to fit best. But well, at least EFSA is watching on that, and already the scientific committee published in 2016 a document setting the priority topics on which we have to work in the following years. And one of the priority topics is to develop guidance to evaluate from a risk point of view, or safety point of view, and from a nutritional point of view, the impact of the individual variability, including also the variability of the individual microbiota that could affect the glycemic response, the nutrient absorption, and also the metabolism of toxic compounds. And now we know, because of the reference and good studies that have been published in the last years, that depending on our microbiota, the value, the nutritional value of a food could also change. And well, as I told you, the classification of these personalized foods is still to be decided because this is not yet there. But of course, we could think that could be conventional foods if we narrow down what we consider subgroups of the general population, of course, and also they could be within the foods for special groups or for a special medical purpose. But as you know, in this case, the food should meet only nutritional needs. Well, regardless this regulatory context and the uncertainties we still have in this field of personalized solutions, what is obvious from the literature and the investigations that are being developed by many groups, I would like to show you how we are moving towards holistic and more personalized nutrition and healthcare approaches. And in this study, the authors show that the glycemic response to a food changed substantially from one subject to another, and that one parameter does not explain these changes, and it's not a good predictor for these changes. For example, the caloric content of the food or the carbohydrate content of the food, and that is only when we integrate all the factors such as the anthropometrics of the subject, the biochemistry of the subject, and the gut microbiota, and we apply an algorithm integrating all these factors, is when we can get a good prediction of the glycemic response of the individual in front to a food. And this could lead us to develop more personalized dietary recommendations, because not all foods will be healthy for everybody. And this is the message behind this paper. And well, this is the way the scientific field is moving forward. And well, to conclude, I would like to address one of the questions that the Commission sent to me to prepare this talk that I understood was a critical one, where these microbiota-based foods could be classified or should be classified as conventional or as medical foods. I understand that this is related to the possibility of making claims or providing messages to the consumer about their health properties. From a conceptual point of view, I can say that they can be conventional and medical foods, of course. As conventional foods, and taking into account the regulatory context, we have to consider that these foods should be intended for general healthy population, and to prove the benefits is rather difficult, despite the fact there is some room for interpretation of the legislation, as I told you, and this is done by EFSA mainly. And should affect also risk factors, and in this context we have to do a tremendous effort to identify and validate new risk factors to make this possible. And well, another limitation is that the functions of the microbiota are now not considered as part of the functions of the human body, and this applies also to the metabolites that are produced from the gut microbiota. And this is an area where the scientific community should work harder, because in fact we have to be conscious that we have not proved causality in many of the cases that we are talking about and are published. And it's really needed to prove that a specific bacteria or a microbiota configuration is really necessary to keep us healthy or to increase our resilience to disease, and the same applies to the metabolites. We need to prove that it's real, that a metabolite is necessary for maintaining our healthy status. And well, in the context of medical foods, according to the definition that is currently in place, they can be intended for managing disease, it can be for diseased people, but if you have read the text, these are intended only for meeting specific nutritional needs. And according to the scientific knowledge we are generating, we are also conscious, all of those who work in the field, that these microbiome-based applications can do much more than meeting nutritional needs, and for example, perhaps could help to improve the efficacy of immunotherapies. And well, regardless all this scenario in relation to the regulation, I would like to finish saying that we should really make an effort to strengthen the alliance between the food and the drug area if we really want to contribute substantially to develop a sustainable healthcare system, because it's undoubtedly that lifestyle, nutrition, the microbiota that is the interface in the middle is playing a role in the progression from health to disease. That is the period where we can intervene and we can do something to prevent disease. So I think we cannot afford the possibility of missing this opportunity to contribute to prevent disease in the EU particularly. And that's it. Thank you very much for your attention.